Hello guys, in these slides we are going to discuss the estimation of proteins by Lowrys method. So before to the starting of the explanation, I request you to subscribe to our channel and also look into the other slides which has been prepared and which has been already viewed by many of the members of the channel who are already subscribed, and also the non-subscribed members also following the slides which has been made into a video log presentation. Now discussing the principle for Lowrys method, which is one of the method to estimate the proteins present in the sample. So before to the principle, we have to discuss 3 points or considered to be as the main factors. That is, the method should be very sensitive and it should give the specificity of the reaction, and time duration of the experiment should be very less or minimal. So here a method should be of sensitive if it can detect protein at very low concentrations. So here samples cannot be taken at the large concentration every time. So at the low concentration itself, if we are going to get the results on the identification qualitatively or estimating it to be on the quantitative basis, the experiment has to give a sensitive output. Specificity of a method is determined by its efficiency to detect protein specifically from all other interfering substances. So here, for example, if you are going to consider within the experiment, there will be a principle. Apart from that, there will be a procedure which will be followed, and in a regular principles like in the college labs or that is under undergraduation, let it be in the pre-university or the +2 or whatever the experiments will be done, that will be a what I call it to be as a ready-made experiments, where a ready sample will be given and there the estimation will be done very easily. But in some labs where the raw materials will be used, and from that raw material whatever the sample has been extracted, on that sample the particular experiment will be done. So here specificity of a method is determined by its efficiency to detect proteins specifically from all other interfering substances. So here during the extraction of that particular sample, protein sample, from that raw material, we are going to subject that raw material to different number of the components. So those traces of that components will be present in the sample. Behalf of those components, if the experiment is able to give the or identify the specificity and give the results, that is the very concerned part. Time duration for assay completion and interpretation of the results. So the time duration should be very minimal so that the result can be achieved very easily and early. So here Lowrys assay method or a Lowrys method is going to fit under these 3 factors, main factors of being sensitivity, specificity and time duration. So here Lowrys assay method is one which has been developed during the 1951 by the Oliver Lowry. So here this is one of the highly sensitive and gives accurate results. It detects the proteins at low concentrations of 2 to 5 micrograms. In some lab structured manuals, they are mentioned to be as the 20 to 30 micrograms. So here there is the difference in the acceptance of the after doing their particular experiments. So it is 2 to 5 micrograms also, it is about 20 to 30 micrograms also. So here both the ways the experiment is going to hold good to find out the presence of the lowest concentration of the protein molecule. In this method, the copper ions are reduced under alkaline condition and form a complex with peptide bonds of the protein. This complex then reduces falling CO-Cal2 reagent and results in the change in color to deep blue, and absorption can be measured at 650 to 750 nanometer. It takes about 45 to 60 minutes for assay completion. A compatibility of Lowry assay for samples with reducing agents and other interfering agents and a modified Lowry assay for samples in detergents are available. So a compatibility of Lowry assay for samples till to the samples in detergents are available. It is the other part. So here what this last point from Lowry assay, from here it is a little bit technique and also the history which we have spoken in a short form. So in the next slide we will discuss the actual principle. Here this is the peptide bond in the polypeptide chain which react with the copper sulfate in an alkaline medium to give a blue colored complex. So here if you are going to observe this particular reaction, in this you can see this is the protein. So it is one of the amino acid which is going to react with the copper cupric ions. So here cupric ions are going to bond with the nitrogen molecules. So here it has got reduced by giving rise to the water molecules, and here as this complex forms, the blue colored complex, blue color within the liquids or the test tubes is going to absorb. Further, adding the falling CO-Cal2 reagent, in which there will be presence of the phosphomolybdate and phosphotungstate, so which are going to get reduced because of the presence of the some amino acids within the proteins, that is the tyrosine and tryptophan. So here further whatever the reduced form of the phosphomolybdate and phosphotungstate is going to take part in creating the bluish products. So here two different reactions are going to take place. One is the formation of the colored complex copper sulfate with the amino acid or the peptide bonds, and other is the reduction of the phosphomolybdate and phosphotungstate with the amino acid like the tyrosine and tryptophan, which they are going to reduce the phosphomolybdate and phosphotungstate. Because of that the bluish products are going to form, where will react with any reducing substance. So here whatever the phosphomolybdate present in the FCR, that is the falling CO-Cal2 reagent, is going to react with any kind of the reducing molecule or any kind of the reducing component, so to give rise to the different component and give the results within that experiment. It therefore measures the total reducing capacity of a sample, which contribute towards enhancing the sensitivity of this method. Here it is in the sense that whatever the amount of the sample is present within the given solution or the medium, that is going to get reduced, and that amount is accurately can be measured further within the procedure of the experiment. So this is what the principle. Now here materials and reagents required. So here first is the phosphate buffer, next is the 20% TCA, again is the alkaline sodium carbonate reagent, again is the copper sulfate reagent, again is the alkaline copper sulfate reagent, falling CO-Cal2 reagent, bovine serum albumin, acetone, 0.1 normal NaOH. So these are the reagents required. So here phosphate buffer 20% weight per volume TCA, and these two are very specific within the procedure, which I will explain in the following procedure. Here alkaline sodium carbonate reagent, here we have to dissolve 2 grams of sodium carbonate in 0.1 normal NaOH and make up the volume to 100 ml with 0.1 normal NaOH. Here this is for only the alkaline sodium carbonate reagent to develop only the 100 ml of the compound. If you want to develop for the 1 liter of the compound, you have to take the 10 times of the sodium carbonate in the 0.1 normal NaOH and you have to make up the volume to 1000 ml. Here copper sulfate reagent is prepare 0.5% of copper sulfate 5Na H2O in 1% sodium potassium tartrate solution in 1:1 ratio. And further, alkaline copper sulfate will be prepared with the help of the alkaline sodium carbonate reagent, copper sulfate reagent. So further, here we are going to, in this we are going to take add 1 ml of copper sulfate reagent to 50 ml of alkaline sodium carbonate reagent. Note this reagent is unstable, has to be prepared fresh instantly during experiments. So you cannot stock after preparing the alkaline copper sulfate reagent. Further is the Fallin CO-Cal2 reagent. We have to dilute this reagent appropriately so that it's 1 normal in respect of its acid content. So here in some lab experiments it will be developed or diluted at 1:1 ratio, but at maximum it will be diluted at 5:11 ratio. So here with distilled water. Further is bovine serum albumin 0.1 gram per liter. Solution is made with distilled water. Acetone is to precipitate proteins, which is required. 0.1 normal NaOH is required. These two components will also be discussed with these components. So here before going to the further procedure, we will have a small talk on this slide where we are going to discuss the extraction for sample protein from raw materials. Here this particular slide is also a part of the procedure, but I have made it little separate with different topic, giving this to be, because in the further slide we will discuss a regular lab experiment. So if this is going to be separate, the learners or the students can understand very well. So here this slide explains how to extract the proteins from the raw material. Here in the labs during the experiments there will be the different proteins which are already present, which are ready-made proteins given to the students as the unknown samples to estimate the presence of the amount of the protein within that, particularly with the Lowrys method. So in some labs or in the higher studies you may get this kind of the situation or the lab experiment where you are going to take this as one of the first step within the procedure. So for extraction of protein sample, the sample can be extracted from germinating seeds, leaves, etc., where we are going to weigh it around 1 gram of sample. Then we are going to macerate or homogenize the sample in pestle mortar in 5 ml of phosphate buffer. So whatever the reagent we have spoken about the phosphate buffer, here we are going to use it. Have enough amount of the sample collected by performing above step repeatedly. So here we are going to do this macerate and homogenizing the sample in pestle mortar or the homogenizer with the help of the 5 ml of phosphate buffer repeatedly, so that the enough amount of the sample got collected. And you are going to combine the sample in one container or the beaker or the large test tube. Then you are going to centrifuge it at 8000 rpm for 20 minutes with the help of the centrifuge tubes. So you are going to further collect the supernatant from all the extractions and make the volume to 50 ml with the help of the phosphate buffer. Here you can see, and add 1 ml of the 20% TCA. So here trichloroacetic acid, here you are going to use 20% TCA. Keep it for half an hour of incubation at room temperature. Then you are going to centrifuge again at 8000 rpm for 20 minutes and collect the pellet formed. Wash the pellet with acetone twice and again centrifuge. Each time discard the supernatant and retain the whatever the pellet is formed. Dissolve the pellet in 5 ml of the 0.1 normal NaOH and mix it well till it gets dissolved and use it as an unknown sample in the experiment. So here further we have come across the use of the phosphate buffer. Phosphate buffer will be helpful to degrade the whatever the cell wall or the cell membrane is present, and it is going to help to break out the cell and release the whatever the proteins present in that. Further you are going to centrifuge, homogenize and centrifuge it, and further you are going to collect the supernatant here. At the first time you are going to collect the supernatant and you are going to discard the debris, whatever the debris has been settled, and whatever the supernatant you got, that you are going to take and you are going to again subject to the phosphate buffer with the help of the 20% TCA. TCA will be responsible for the precipitation of the proteins. So here you can see TCA is used to precipitate the proteins, and further you are going to keep it for incubation. Incubation here is nothing but you are going to give the time for the chemicals like the phosphate buffer and the TCA to show their complete reaction on the molecules or the biomolecules for which we are concerned to do the experiment further. So further keep it for half an hour in incubation at room temperature. Then we are going to centrifuge at 8000 rpm for 20 minutes and collect the pellet formed. Wash the pellet with acetone twice and again centrifuge. Each time discard the supernatant. So we are going to wash the pellet with acetone. Acetone is also a kind of the protein precipitant, so it is going to help very purely to precipitate the protein. Further you are going to dissolve the pellet in 5 ml of 0.1 normal NaOH and mix it well till it gets dissolved and use it as an unknown sample. Now here NaOH is going to keep the proteins in their particular natural form or in their particular structures. So that is what here, why here we are going to dissolve the pellet into the NaOH. Now further is the note. There may be interference in color development by some products using the experiment. Those can be like EDTA, trace carbohydrate, NH4+, K+, potassium, magnesium, etc. Hence has to be careful and avoid this. Here, as I have spoken earlier, that these chemicals, if they are at the high concentrations, they may act as the interferers and cause the experiment not to be up to their sensitivity or specificity. So this has to be removed very well before to get into the actual procedure of determining the or estimation of the proteins. So here within this whatever the procedure, we have done, the maximum to maximum amount of these molecules will be removed when you are going to add the acetone and you are going to subject within the NaOH. So this is going to remove, and at the last centrifugation twice we are going to do this centrifugation. So because of that, there will be the removal of the maximum amount of these traces of the chemicals. So we no need to worry about this particular part, but provided you have to do the experiment very carefully. In this slide, we are going to do the procedure with the help of the standard stock solution of the bovine serum albumin and the sample what we have extracted. Now here in the regular labs, there will be no extraction from the raw materials will takes place for either it can be in the undergraduation or the pre-university or +2. In this, during that time, the students will be given with the ready-made proteins dissolved in the test tubes or prepared solutions to identify the whatever the amount has been given to them. So here, before to that, we have to understand the bovine serum albumin stock solution. So here we have gone through this. Bovine serum albumin will be taken as 0.1 gram per liter. Solution is made with distilled water. So here, this stock solution, which is having 0.1 gram, it means it is going to have 100 milligrams. So 100 milligrams is equal to 1 lakh micrograms. So here, when you are going to consider each ml of the bovine serum albumin stock solution of 1 liter is going to have 100 micrograms of the bovine serum albumin. It means that if you are going to calculate this 1 lakh micrograms for 1000 ml, so here you are going to get 100 micrograms per ml as the result within the bovine serum albumin. 1 ml is equal to 100 micrograms. So here this is explained because further within the procedure there will be a doubt. To clarify that, now itself this has been spoken out. Now here, from that BSA stock solution, we are going to prepare standard sequence liquids like 0.2 ml, 0.4 ml, 0.6 ml, 0.8 and 1 ml. This particular whole slide I will discuss here in this protocol. In this, you can see that we have to take 6 test tubes plus 2 test tubes of sample 1 and sample 2. So total of the 8 test tubes. Here we are going to take around 1 as the blank, second as the 0.2, 0.4, 0.6, 0.8 and 1 ml as the standard BSA liquids in ml. Further we are going to take sample 1 and sample 2 of 1 ml each, which we are going to estimate further after the completion of the experiment. The concentration containing within this standard BSA liquids is for 0.2 ml 20 micrograms of the BSA, 0.4 ml 40 micrograms of the BSA, 0.6 ml 60 and 0.8 80 micrograms, for 1 ml 100 micrograms. So the samples has to be estimated. Further we are going to make up the volume of this lesser than 1 ml standard liquids to 1 ml by adding the respective amount to be added to make it to 1 ml. For the blank we are going to add 1 ml as the blank is not having any of the solution or the sample added. So further, after doing this addition of the distilled water, we are going to add volume of 4 ml alkaline copper sulfate reagent in each test tube. So all the test tubes, from the test tube 1 to the test tube 6 and sample 1 and sample 2, we are going to add the alkaline copper sulfate reagent in each test tube, and we are going to shake it well or vortex it well, and we are going to keep it for incubation at room temperature for 10 minutes. And further, we are going to take the test tubes and we are going to add further 0.5 ml of the Fallin CO-Cal2 reagent into the each test tube, and further we are going to keep it for incubation again at room temperature in the dark for 30 minutes. So further, after the 30 minutes, we are going to take the test tubes and we are going to see the absorbance in the photocalorimetry or else the spectrophotometer at 660 nanometer. So first we are going to fix the blank within the spectrophotometer or the calorimeter, and further we are going to take the readings correspondingly to the liquids, and these are the readings what we have got from here to here last. So here again, this we are going to plot on the graph. Now here you can see that the graph has been taken accordance to the concentration of the BSA in micrograms to the against to the or the versus to the absorbance at a 660 nanometer, absorbance at 660 nanometer. So you can see this will be the standard curve, this will be the absorbance unit, this will be the concentration of the BSA in micrograms, what is the unit on the x-axis, this will be the unit on the y-axis. Now here, as we have observed that the samples are having 0.24 and 0.23 as the absorbance reading, that has been interacted here. 0.24 has been considered, a single sample reading has been considered, so that has been considered here, and it has been interacted on the standard curve, so that is going to fall to this particular point on the graph for the BSA in micrograms. So here in the graph it is showing it to be as the approximately 70 plus micrograms of the protein is present in the sample solutions. So further we are going to consider these particular readings what we have got, and we are going to calculate in the formula. So here what we have got is these readings. Now that we are going to consider in the formula. So concentration of unknown is equal to absorbance of unknown divided by the absorbance of standard multiplied by concentration of standard. So we are going to subject all these values to know the concentration of the unknown. Concentration of the unknown is nothing but the proteins, estimation of the protein which is present in the sample, that is the unknown sample. So that here according to this, absorbance of unknown is here 0.24, that is the last reading. This will be the absorbance of unknown, that is 0.24 here, and absorbance of the standard is 0.20. here absorbance of the standard is 0.20. So it is nothing but this one, which is near to the 60 micrograms. This is going to fall here. So concentration of the standard, absorbance of the standard, concentration of the standard 60, what we have seen in the previous slide. We are going to calculate concentration of the unknown, which is to be estimated. Now after subjecting all these values within the formula, we get 72 microgram per ml of proteins in the sample. So this is what the accurate result which we have got in this Lowrys method. Further, we request the viewers or the followers of our channel to subscribe to our channel and to help us to provide you with the more information on different topics, and also help us to by giving a specific topic which has to be discussed, and also help us by giving a deliberate feedback how far it is good and how far it is has to be improved. And also I request everyone to spread the word of these particular slides, and also be tuned on Big Book.